I think one of the things that most of you have heard about, or at least spent time in the ocean, is we think about the big stuff. But what truly matters is the small stuff. So we're going to spend a lot of time talking about the small stuff of the ocean that you don't get a chance to see. And many of you probably know this, that the most eloquent writings on what happens in processes in the ocean in the past have been written by a remarkable author that some of you know about, Rachel Carson. And one of the things that she said in her writings, she was a marine biologist by training, and this is what she wrote. The ocean is a place of paradoxes. It's a home to the great white shark, the 2,000-pound killer gorillas, the blue whales, and then goes on and on. But then she also talks about what truly matters is life that you can just hold in your hand and that drifts endlessly into the depths of the abyss, what we call plankton, the wanderers. And what I'm here to show you is for the last 200 years, I think we have gotten this wrong. Plankton are not wanderers. And it's incredibly important to understand what they actually do. So this is our planet. It's not just a blue ocean. There's an incredible amount of life. If you were to shut down what happens in the ocean from a biological context, which is called the biological pump, the CO2 in our atmosphere at this very moment will go from a 400 to 600 parts per million. You might wonder how. Those are the mountains of Dover that are made out of individual cells that are 5 micron. And that's literally carbon sequestration that's happening. When life dies, it sinks to the bottom of the ocean, locking it to thousands of years. Now, how do we study this? This is one example of a dive, and this is the density of life that you can actually find. And what I want you to imagine is if you could cut a chasm into the ocean and actually see along this vertical axis what's happening. What are the cells doing over a function of time? Now, in the World War, it was discovered with a submarine when they realized that the ocean floor rises and falls every night, which freaked a lot of naval officers out. It turned out it was actually life that was bouncing the sound signal back. Everything in the ocean is actually vertically stratified. Everything from just 100 meters, 200 meters where light is. You want to see photosynthetic cells there. Falls all the way down. Every parameter, property, salinity is actually stratified this way. Now, the challenge is that ecosystem spans 4 kilometers, and we have to try and understand life, even a single cell that has the possibility to travel hundreds of meters in a single day, except we ran into a challenge. So first of all, this is where we stand today. This is literally a title of a paper. I'm not going to say what the bleep is wrong with our present calculations of carbon budget. The problem is we put biology in most of our models as a spherical cow. We really don't understand the context of what the cells are actually doing. And this leads to massive disconnect between what you can measure from the atmosphere and what you can directly measure in the ocean. So here is the problem. We've been thinking about this. How do you even watch a single cell that could freely travel of hundreds of meters? I proposed this idea. We're going to make the world's tallest microscope. That didn't go well with my chair. But then we came up with a better idea, which is to build a treadmill. So what we've built is an ocean on a tabletop, which is, we call it a gravity machine. A couple of tricks that we play, but the simplest way of thinking about is think of a wheel, and when the organism is moving down, we move the wheel up. When the organism is moving up, we move the wheel down. But for the first time, we can actually observe cellular processes of freely swimming single cells that are traveling infinite distances on the vertical axis. We've been, for the last 3 years, off on many boats. This is one example of where we run the experiments. Just to give you a flavor of some of you who have never worked on a boat, this is data set of a cell that we collected 300 meters deep into the water. We brought it to the machine in a pressurized manner. We're collecting its behavior in situ at that time while there was a storm coming. And actually 2 hours after this moment, we had to turn the ship around because it was too dangerous. That's the storm. Now, the types of discoveries that we've actually made is one of the key things we're studying are pandemics in the ocean. We have realized that viruses play an incredible role in how phytoplankton blooms die. And what that would mean, this is work in the Mediterranean with my incredible collaborators. We have discovered how carbon actually fixes. This is the first video of marine snow which is really our carbon flux. But now you're falling in the frame of reference of this while it's falling 200 meters, and we can try understanding how these structures change over time. We've discovered several analogies of completely bizarre geometries of cells. So for some of the topologists and the mathematicians, this is a cell that's an N-torus. For the first time, we're discovering a cell that is not just a sphere. It actually is topologically incredibly complex. And the reason this topology is important is because the cell uses ballooning as a criteria for essentially rising and sinking hundreds of meters in a function of a day. This movie is going to play. Maybe it's not going to play. What you would see is this massive expansions of these cells over time. And this is where topology comes into play, because you cannot just dilute the cytoplasm. I'm going to end with this video, which is, for me, the most remarkable video that I've ever collected out in the field. Actually, can you play this video? If this video was to play, no, this is not. This is a beautiful video. This is in the context of sinking and swimming with starfish larvae. But the video that I actually want to show you is the diatom video, which is one below. What we ended up discovering is cells that we thought are immobile and immotile, have no cilia, no flagella, have the capacity to change their density in hundreds of milliseconds. So this would imply that if I was to drop literally this mic at this moment right in front of your eye as it was falling, it has the capacity in 100 milliseconds to levitate. This is incredibly important because diatoms essentially produce 50% of all the oxygen that we breathe. I'm going to end this anyway. We're creating a zoo of these organisms. This is literally one of the largest data sets that's accessible of freely moving behavior of life, plankton in the ocean. And then the other part of this is actually sharing much of this work with a broader group of people by building scientific tools that we can give to sailors, because I cannot afford spending $50,000 to $70,000 per day that I actually have to spend being in the ocean. So we're building many of these tools and transforming them to what we are calling citizen scientists. So a broader group of people can actually collect data in the ocean. Thank you.